Yes, so thank you very much, Pauline. So hello, everyone. Welcome to my presentation. As Pauline said, for those who don't know me, my name is Yannis, and I'm a postdoc working in the Lauritz School group. We're based in the Globe Institute in the University of Copenhagen. And my main research interests are in phylogenetics and population genetics, and especially combining those two for the study of human history. However, more relevant to today's talk, I did my PhD thesis, supervised by these two gentlemen, Fernando Racimo and Enrico Capellini, and the title of the thesis was Reconstructing Deep Population History Using Paleoproteomics. And in this thesis, we tried to combine these previous interests of mine, phylogenetics and population genetics, with the emerging field of paleoproteomics. And in fact, the work I will be talking about today was the third chapter of the three chapters of my thesis, and it bears the same title as in the thesis. And this thesis I defended back in 2023. However, it was finally published in its final form more than 2 years later, this last February 2026. So if there's any PhD the talk listening, just know that you might be finished with the PhD, but the PhD is not finished with you when you defend. So the work we'll be talking about is now published in GBE. You can find it using this title. But before I get to it, I want to give you a bit of background and some context to this work. And the background is that if you're someone like me who is interested in studying human evolution, you will know that most of our ancestors lived in very warm places of the world, like Africa and Southeast Asia, and most of them lived millions of years into the past. And this combination of deep time with warm and humid environments means that preservation of DNA for this sample is going to be, for all of these samples, is going to be quite bad. And ideally, this is how we would want to study these samples with DNA. And this is where the field of paleoproteomics enters the picture. This field promises to get molecular data from organisms that lived more than 1 million years ago and from regions of the world that are traditionally unfavorable to biomolecular preservation. So has the field delivered in this promise for human evolution so far? I would say so that yes, and you can take a look around here for all the samples that have been published until now that are human related. I actually had to update the slide a couple of days ago because there are recent arrivals like these Homo erectus specimens from China or the Homo naledi samples that were published a couple of weeks ago. But in a sense, we do get a lot of different proteins from different samples and from a lot of different samples. However, there are still reasons why we should be skeptical about the application of phylogenetic applications on polyproteomic data. And that is because the transcriptome, which translates into the proteome, so the full number of proteins an organism possesses, is a mere fraction of the total genome. You can see how small it is right here. And in humans, it's around 1 to 2%. Additionally, we always never get to work with the full proteome. We usually get a handful of proteins, usually from teeth or bones. And even then, those sequences tend to be very fragmented with a lot of missing data due to degradation. This means that we have a very limited view of the whole genetic history of a species or of a group that we're studying. And you may say, so what? It's better to get some information than to not get any data at all. And I would probably agree with you. However, I do like to give this example of what happens when you only have access to mitochondrial DNA data from archaic and modern humans. So if you had only access to this type of data and you plotted it in a phylogenetic tree, the relationships, you can see here there is a group of Neanderthals, here is a cluster of modern humans, and here we have another cluster of Denisovans and these other hominids from Cima de los Huesos, which is in northern Spain. And looking at this phylogenetic tree, you might come to the conclusion that A, Neanderthals are actually closer to modern humans. You can see it right here. And B, that Neanderthals and humans never actually interbred because they form these perfectly separating monophyletic clades. And actually, a lot of people around 1 or 2 decades ago believed this or were supporting this with their studies. And this was mainly based on this mitochondrial view. However, when we got access to the whole genome data from all these different groups, we could actually see that a lot of these conclusions were wrong. So we could see that actually the Neanderthals are more closely related to this other group, the Denisovans. And in fact, these Cima de los Huesos individuals are ancestors to the Neanderthals. And also that Neanderthals and modern humans have interbred both a couple of hundreds of thousands of years ago and again, a couple of tens of thousands of years ago. In addition to this, most mutations on genes are silent or synonymous, meaning that they happen on the DNA level, but they don't cause a change on the protein sequence itself. And also proteins are in generally under constant selection, meaning that their sequences are very conserved even across species. And as an example here, I have some sequences from histone H1 from different species, so humans, chimpanzees, rat, a cow. And you can see that even as things as this a human and a cow have 95% similar sequences for this protein. That means that there are very few sites to work with when we're trying to do phylogenetics or even things like species identification. And usually we're focused on these sites, like the one right here, where there are differences between the different species or the different groups. Right, so with all of that in mind, the year is around 2022. I'm doing my PhD thesis, and Fernando and Enrico decided that one of the chapters of the thesis should be testing how bad, or you want to say how good, ancient protein sequences are for doing this type of phylogenetic analysis. Until that point, we had seen that ancient proteins do contain some phylogenetic signal, but provide very low resolution for resolving relations among the very closely related species. And this was noted explicitly in multiple previous paleoproteomic publications. I put here a couple of them. But it was still unclear how much phylogenetic information is in these deep time proteins, and how much our evolutionary inferences using these proteins will be affected by things like incomplete lineage sorting or admixture. And you may be asking, how do these things like incomplete lineage sorting affect our evolution inferences? Or you may simply be asking, what the hell is incomplete lineage sorting? And for this, I have this little figure that I can show. Imagine a population tree between humans, chimpanzees, and gorillas. We know that humans and chimpanzees are generally closer to each other than they are to gorillas. So we expect most regions of the genome to have a gene tree that looks like this, with the human and a chimpanzee being closer to each other and sharing a mutation. Let's say they share an R. And then the gorilla being an outgroup and having a different mutation. Let's say having a Q here. And we can imagine that most of these mutations happen either in the lineage leading up to modern humans and chimpanzees, or in the lineage leading to gorillas. However, there are regions of the genome that will have a gene tree that looks like this, with a chimpanzee and gorilla sharing a Q mutation and the human having a different mutation. And the chimpanzee and the gorilla will appear closer to each other. And this is where this can happen when there is a mutation in the common ancestor of these three groups right here. And then by random sorting, chimpanzees and gorillas get one mutation, whereas humans get the other one. This is what we call incomplete lineage sorting. And incomplete lineage sorting is more likely to occur. I can also use ILS for short. Sorry, I think someone has the microphone on. But ILS is more likely to occur when this population right here is very large in its population size, or when the splits between these different species happen very close in time, and then it's more difficult, or the sorting is more random. Of note, even in regions where there is no ILS, like the one right here, one might still see chimpanzees and gorillas appearing closer if there was an ancestral mutation that is a Q, and then a mutation happened at the very end or the terminal branch of the tree leading to humans, causing them to have an R, and causing these ones to look closer to each other when they are actually not. Finally, a different genetic phenomenon known as admixture or interbreeding can also cause the same issues. For example, imagine there was a mutation right here into the gorilla lineage, but then somehow chimpanzees and gorillas interbred. And then chimpanzees ended up having the Q mutations with the gorillas and appearing closer to them on this specific spot. So in conclusion for this, ILS, admixture, and recent mutations can all substantially alter our evolutionary inferences. And again, just a summary, we wanted to investigate how much phylogenetic information is these proteins, how much our inferences will be affected by things like ILS and admixture. And we actually decided to focus on hominids for this project, which are the closest human relatives. And this, of course, because we are humans, we are interested in our own genetic history and that of our closest living relatives, but also because studying hominids can be a bit challenging. For example, multiple studies in the past have identified that up to 30% of the genome of great apes is undergoing ILS, which is what we just saw. So humans, chimpanzees, and gorillas have many positions on the tree where chimpanzees might appear closer to gorillas, or humans might appear closer to gorillas. And also from ancient DNA, we also know that the late hominids, at least, like Neanderthals and Denisovans and our most recent ancestors, modern humans, admixed a lot. And pretty much everyone has admixed with everyone else, leading to many parts of the genome that originate from another group rather than the original that we're studying. And what type of data are available to investigate these type of questions? Well, we were lucky because we had data from a previous publication of ours where we created this hominid paleoproteomic reference data set and actually had a PAASTA talk, I think it was more than a year ago, that I talked how this data set was created, and it's available on YouTube if you want to watch it in the PAASTA channel. But importantly, this dataset contains representatives from all extant hominids, so chimpanzees, humans, gorillas, and as well as two extinct populations in the Neanderthals and Denisovans. And more importantly, contains proteins of our interest. So for this project, these were 12 proteins that have been previously recovered in samples that are older than 1 million years. And here I colored them with either blue, if they belong to enamel, or with red, if they belong to collagen. And of course, this was a couple of years ago, so things might change and we might recover more proteins as time progresses, but this is what we had at that point. So with this dataset, the first question we try to answer is how much information lies within these proteins, which is another way of saying how conserved these proteins are phylogenetically or evolutionarily. And for this, initially, we tried using this metric called Shannon's entropy. And this metric can tell you how much information is in the protein alignment, with a less conserved protein having more phylogenetic information, and thus a higher entropy. And we can see here a couple of examples. It is a bit confusing. Here I have an alignment of different species and groups close to humans. So every row here is the protein from a different species aligned with the other ones. And then we can see for a position like that one of 636 for collagen 17A1, some species have an A, where other species have a P. So this position has some entropies, has some information, and thus gets an entropy score of 0.8. Whereas other positions, like this 638, or many of the other positions in this alignment, actually have no diversity in their alignment. It has no information, and thus has an entropy of 0. Another example here, another protein, albumin, again, alignment in different groups. Here we have 4 different observed states on position 183. So we have an E, we have an A V, or a K. So this position has a lot of information, a lot of entropy, and gets a higher score of 2. So what we did then, we made alignments for our 4 hominid groups, Pongo, Gorilla, Pan, and Homo, for all of these 12 proteins. And then we summed the entropy of each position across the alignment. So imagine we have this alignment for these groups, and then most positions have a 0, but this one has a 0.8, as we said. So this alignment in its sum will get a 0.8. And of course, you can imagine that if it expands this way, you will find more positions with entropy, so the alignment sum will increase. And then we compared this sum with known conserved proteins like histones and ubiquitins, which we know from basic biology that they should be very conserved because they are very important to an organism. And also known variable proteins, like we found these fibrinogen proteins from the literature that supposedly have a segment that is hypervariable. And then we plotted this aggregated entropy for each of our protein alignments. And you can see here the results sorted by the aggregated entropy on the right. And the first thing we saw was that our enamel proteins have a wide range of entropy. So some proteins like enam have a lot of entropy, where other proteins like AMLX have very low entropy. We also saw that most of our enamel and collagen proteins were had more information than the very conserved proteins or ubiquitins and histones, which you can see here at the very bottom with purple and yellow. And also these other fibrinogen proteins were kind of in the middle of our distribution of entropies. However, we noticed that it was the longest proteins that tended to have a higher entropy, like enam and collagen 17A1, in theory, are very long and thus have a lot more chances for mutations to occur. So we decided to account for that, and then we divided each of these entropies with the length of the protein. And you can see this changes the picture quite a bit. First of all, we see that now our NAM protein and our collagen 17A1 have dropped significantly, in the distribution and are now kind of in the range of distribution, whereas other proteins like ORAM and AMTN, which are usually quite shorter, actually have very high entropy. And this is because they have a lot of information for their length. Finally, we saw that our collagen proteins actually drop to the very bottom of the distribution when accounting for their length. And they're pretty much as conserved as histones and ubiquitins, so they don't have that much information. And originally, this was the end of our analysis. However, we did have some comments from one of the reviewers, and I think one of the editors or the collaborator, which said, oh, you should use an additional information metric. And for this, we tried to use this evolutionary rate metric, which actually was used in the previous analysis by Fong, Zazueta et al., which I also encourage you to look at if you're interested in what I'm doing here today, because they did very similar things. So this new metric, the evolutionary rate, is an approximation of the speed at which a molecular entity, such as in our case a protein, but also could be a gene or other molecular entities, evolves. So how fast they accumulate mutations. And for practical reasons, we use a program called rate4site, which can generate you those metrics for an alignment. So essentially, we put the same protein alignments into rate4site, and it gives us the evolutionary rate. And here I plot side by side the evolutionary rates and the entropy rates. And we can see that in most cases, like which proteins have the highest evolutionary rate or entropy, they agree. So NAM and collagen 17A1 are the highest. But other cases, like the collagen type 1 proteins, there is some disagreement, like evolutionary rates based on a bit higher in distribution than the entropy. However, when we again divide it by the length of those proteins, we actually see now that our two different rates or metrics agree mostly with each other. So ODAM is very high in both cases. Then we have this cluster of AMLY, HSG, and AMTN here, the same. And then again, the collagens seem to drop significantly and are on the lower side of our distribution. So almost as conserved as the histones and ubiquitins. And again, also something other I would point, AMLX is consistently showing very low. Even if it is a short length, it is super conserved for its length, and thus it's pretty much as conserved as one of the collagens or the histones and ubiquitins. Whereas something that I would also point is AMLY is actually quite more diverse than AMLX, each X chromosome equivalent. So as for the results of our first question, we saw that the proteins with the most information were NAM and collagen 17A1. But after accounting for the protein length, we saw that ODAM, AMTN, and AMLY were some of the most variable enamel proteins. And collagen type 1 and AMLX were some of the most conserved proteins that you could get for these very old samples. With that, our next question had to do with informational loss. We know that there is information loss when moving from DNA data onto protein data. For example, DNA has 64 different DNA codons, so these are triplets of DNA bases, which only get translated into 20 different amino acids. And this also has to do with the silent mutations. As I said, these are mutations that happen on the DNA level, but do not cause a change on the protein level. So it is mutations that they were essentially are invisible to us if we're only working with proteins. So we wanted to see whether this informational loss or drop from DNA to protein data impacts the inferred gene tree topology. And you may say, how would that impact? Well, you can imagine again, we saw that there are these regions of the genome where the gene tree, the local gene tree, differs from the population tree. So this tree right here is different from the population tree. And this could be either due to admixture, ILS, or recent mutations. However, we usually expect the protein tree to match this gene tree and thus being different from the population tree. So this one way protein trees can differ from the population tree. However, it could be the case that the gene tree does match the population tree. So this local region or gene has a population history that matches the population tree and gives us accurately which tree it is. However, it is the protein tree that actually differs from the gene tree and thus differing from the population tree. So we want to get a sense of how often this happens. And to investigate this, we use alignments again of proteins, but this time in conjunction with exon data for these 12 genes and 12 proteins. And again for Pongo, Gorilla, Pan, and Homo. And then we generated a tree using each one of these alignments. So we had 12 genes times 2 for our 2 different data types, DNA and proteins. So 24 trees in total. And then for each tree, we assigned it a topology among these 4, topology 1, 2, 3, or 4, where topology 1 is the one that we know matches the population tree, with humans as an H, matching being closer to chimpanzees with a C, and gorillas being the outgroup. So this is the population tree as well. Whereas other topologies like 2 has humans and gorillas being closer, topology 3 has gorillas and chimpanzees being closer. And finally, topology number 4 is called the polytomy because actually we don't have enough information to tell which one of these species is closer to one another. And then we plotted our results, which trees from which data type match which topology. And we saw that most trees, either from DNA or proteins, do create this topology that matches the population tree, with humans and chimpanzees being closer. So if you're using ODAM, if you're using DNA or proteins, you will get this tree. If you're only using collagen 1A1, either proteins or DNA would get you this tree. However, we saw 2 cases out of the 12 where the DNA inferred gene trees are different from the species tree. So with NAM, if you're using the gene, you actually get this species tree, which has humans and gorillas appearing closer, same as AHSG. And then finally, we saw that 5 out of the 12 proteins, so almost half of them, had a protein inferred gene tree that shows a topology different from the species tree. So if you're using the protein of MMP20, you will get a tree that has humans and gorillas closer. If you're using collagen 1A2, the protein, you will get gorillas and chimpanzees closer. But if you're using DNA, you will get this tree. And finally, we also saw with AMLX, there is not enough information for the tool to make a phylogenetic prediction. So we get this topology, which is a polytomy. So we cannot infer which one of these species is closer to which one. However, we saw that proteins differ from DNA trees of the same gene, which we kind of expected because of the previously mentioned drop in information. And this is not very surprising. And we also know that individual gene trees can often differ from each other and from the population tree of a species. And again, I would recall to the example of humans and the adults and denisomans. When you're looking at the mitochondrial, you would get a very different tree from what you get if you look at the whole genome or other parts of the genome, either due to admixture or ILS. But what was more interesting to us is what happens to the inferred tree topology as we concatenate more and more proteins. Theory says that if you add more and more proteins, you should get closer to the population tree. So we wanted to test that and see how that goes. So for this question, we chose to test the relationships of 2 different data sets. One was our previously used Homo, Pan, Gorilla, and Pongo, which I would classify as an easy to medium level of difficulty tree. Medium because, as I said, there is some ILS between these different species and completely in sorting. And then we also had this additional data set of Neanderthals, Denisovans, modern humans, and then using chimpanzees as the outgroup, which I would classify as a very hard data set because these are very recently diverse groups. A lot of people don't accept them as being different species. We just call them as different populations of the same species. And also we know that they have admixed a lot in recent times. And initially we restricted ourselves to using, again, these 12 deep time proteins. So for this, it's a bit difficult to follow, but I will do my best to explain what we did. We designed an iterative analysis to resolve this question. So for this iterative analysis, we picked an n between 1 and 12, where 12 is the maximum number of proteins that we can use, and 1 is the minimum. So let's randomly give an example with n equals 3. First, we randomly pick an n number of proteins from the 12 that are available to us. So for an n equals 3, we pick randomly AMLX, NAM, and AMBN. Then we randomly pick 4 individuals, 1 from each of our populations from our data set. So a random individual, let's say this time it happened to be a human from Iberia, a random Pan paniscus, a gorilla beringae, and a Pongo belly. And then we create an alignment for each of these 3 proteins, so 3 alignments, and then join these 3 alignments into what is called the concatenated alignment of 4 individuals and 3 proteins. And then finally, we generate a phylogenetic tree, record its topology among these 4. Again, the 4 topologies we saw for humans, chimpanzees, and gorillas. And let's say re-record the topology number 1. And then repeat this whole process 1,000 times, each time picking different set of proteins, different set of individuals, and then redoing the phylogenetic tree and recording the topology. Finally, we do this from an n1 all the way to 12. So we end up 1,000 times for each n, so 12,000 concatenated trees. And then we plot the results. And here is the figure that shows the result. I'm going to show a bunch of them, so I'm going to spend some time explaining them. In this top row here, we can see the number of trees disagreeing with the population tree, so topology number 1, over the number of proteins we use. So going from 1 all the way to 12 proteins, how many of the 1,000 trees we generated disagree from the topology that has humans and chimpanzees closer. And then on the lower, right below it, we have out of the 1,000 trees, again from 1 to 12, the full range or proportions of phylogenetic trees we generated among these 4 different topologies. So these 4 different topologies have 4 different colors. And then we paint this 1,000 tree bar plot with the percentage of trees that we generated. Finally, this lower part of the figure has the number of informative sites that were generated, were found in the alignment that was used to generate a tree. So you can see here on the very left, when we're using one protein, we have a bar plot here, box plot, sorry, that goes from 0 to 25 informative sites when you're using one protein. Whereas when you're using all 12 proteins, you can have an average of, let's say, 100 and something informative sites. So this is essentially counting how much information our phylogenetic tree used to be generated. So with this in mind, for the hominid dataset, let's see some of the results. First, we noticed that as the number of proteins increased from left to right, from 1 to 12, we saw a steady increase in the number of trees supporting topology number 1, which again, I will remind, is the one with humans and chimpanzees being closer and gorillas the outgroup, so our population tree. And you can see it with this purple bar plot, which is steadily increasing when using more and more proteins. And another way to see it is this bar plot steadily decreasing. So these are the trees disagreeing with this topology as we're using more and more proteins. And you can also see roughly how many of informative sites were used for these phylogenetic trees. And then we saw that with around 4 proteins on average, we could eliminate the polytomy. So this is the black topology, which is when we cannot tell anything about our the relationships between our different species. And with around, let's say, 8 to 10 proteins, we could consistently get the tree with humans and chimpanzees being closer, which is what we want. This is the population tree as we know it from other forms of data. So in conclusion, for this first data set, the hominid data set seems to be pretty good, and things that are as related as the hominids are in our data set should be able to be differentiated with a relatively low number of these conserved proteins. Now let's see the results for our hominin data set, where the picture is quite different. Here, we actually saw that as the number of proteins increased, we only saw a minimal decrease in the trees disagreeing with topology number 1. So this is the topology with Neanderthals and Denisovans being sister groups, and then humans, modern humans being the outgroup, which is what we know from DNA is most of the genome. And then you can see there is a slight drop as you're adding more proteins, but then it kind of plateaus. And even in some cases, it goes back to being more trees disagreeing. And in fact, we saw this purple, again, slightly increasing, but then it stagnates. We saw this other topology with darker blue. So this is the topology with Neanderthals and modern humans being closer and Denisovans being the outgroup, steadily increasing as you added more of these 12 enamel proteins. Additionally, we saw that the polytomy, so the black topology, was never fully resolved. Even when using 12 proteins, you see a slight percentage of your trees generate topologies that look like this, where we cannot tell anything about the relationship between our different groups. So our initial conclusions from this was that these populations are too closely related with one another, and that true ILS and admixture might be affecting our results, and that we will need additional proteins to recover the population tree for these very closely related populations. And originally, this was the end of the whole project or an analysis, but we did get some suggestions from some of the reviewers. Specifically, they said to look about what happens when you use more proteins. For example, we know that if you're using dentine, you might get more proteins than just the collagen type 1. And what about what happens when you try to control for admixture? We know that Neanderthals, modern humans, and Denisovans have mixed a lot. surely there are ways to control for this admixture and test if it is the reason why you're seeing these results. So for our first question, what about using more proteins like dentin for hominids? We chose 12 proteins that were identified in a previous publication, Ruther et al. 2020, as being the 20 most likely to be recovered from bone and dentin. So we added our two datasets. Unfortunately, 4 proteins were overlapping between these two datasets. 2 of them were collagen type 1. I think the other one was collagen type 2, and there was one more bone protein. So in the end, by joining these data sets, we only have 28 proteins. And then we acquired again the proteins, these 28 proteins for our human, Neanderthal, and Denisovan groups. And we redid the iterative analysis, this time going from 1 all the way to 28 proteins. And we saw slight differences. For one, we saw the steady decrease of trees disagreeing with topology number 1, or the other way, this purple topology, slightly changed, but again plateaued with around 20 proteins. And again, we had this other topology with humans and Neanderthals be closer, Denisovans being the outgroup, steadily increasing, but again, this time plateauing and then essentially being roughly 50-50 with this true population tree as we know it from DNA data. So yeah, with 28 proteins, we have roughly the same support for the topology with Neanderthals and modern humans being closer, and Neanderthals and Denisovans being closer. And luckily, we were also able to show that, with around 20 proteins, you're fully able to say something about your samples, about the relationship. So, you need around 20 proteins to be able to tell that. And of course, something I forgot to talk about in the previous plot, the number of informative sites that they're using goes from 0 to, well, maybe a maximum in some cases of 50 informative sites, which if you remember from the hominid dataset is not even half of what we had for 12 proteins. So this is something to keep in mind. For the other question of what about admixture affecting our results and how to test this, luckily at that point I had moved to a new group as a postdoc with this guy called Ioannis Patramanis. I actually forgot to put his name here, but this is him, who is an expert in admixture between the late humans, Neanderthals, and Denisovans. So I was quite lucky. And with him, we designed a way to test this, whether or not admixture is affecting our results. First, we wanted to test if any of these 12 or 28 proteins fall within regions of the genome of modern humans that are known to be introgressed from other archaic humans. So we know a small fraction of some humans has Neanderthal DNA, and a small fraction of some humans have Denisovan DNA. So we want to see if proteins fall within that tiny fraction. And then we also wanted to see what happens if you include in our iterative analysis only modern humans that have very little admixture. So we know that sub-Saharan African people have been inferred to have very little Neanderthal and Denisovan admixture into them. So for the first part, we looked at 2 studies that have performed introgression scans on modern human samples. So these are samples of the 1,000 genomes or the GTP dataset. And these 2 publications are Skov et al. 2018 and Chen et al. 2020. And then we counted how often do we find any of our 28 proteins within these introgress regions as they have been inferred from these introgression scans. And we saw that a number of our enamel and dentin proteins fall within these introgressed regions. So we saw that about 9 out of the 25 proteins fell within these regions. However, most of the cases, they have a very low frequency when you're looking at the global human population. Usually it's around 1 or less than 1% of humans that have it. And unfortunately, we could not look at amelogenin X and amelogenin Y or BGN because these are on the X and Y chromosomes, which in a lot of these genome-wide scans, they avoid the Y chromosome or the X chromosome. The Y chromosome is because we have no data that show that the Y chromosome has introgressed in any modern humans from archaic humans. And the X chromosome has some technical difficulties with inferring introgressions. So most studies actually avoid these 2. So this is why we have 25 and not 28 proteins here. And unfortunately, the best way that I could plot all of these results was in the form of supplementary tables that you can evolve. You can see if you're interested in specifically some of the proteins. But here I chose to show some notable exceptions to what I just mentioned. So for the neanderthal proteins, we saw that one of the neanderthal proteins, MMP20, actually has very high frequencies in Europeans, up to 40%, and also in Asians. And this decreases for other populations, but even in Oceanians, it's up to 16% of the population has actually the Neanderthal version of MMP20 based on these introgression scans. And also for collagen 11A2, about 30%, which is the highest in modern humans, is in Native Americans, and it drops for Europeans and Asians, but still relatively high, and very low in Oceanians. On the other hand, we saw some denisovan proteins, particularly ODAM, ALB, collagen 22A1, LUM, and collagen 5A3, that were in 20% or higher frequencies in the Oceanian modern people, which we know from other forms of data that have a lot of denisovan ancestry. So that makes sense that they have a high percentage of denisovan proteins. But this drops to less than 1% for global or other human populations other than the Oceanians. Unfortunately, we're not able to find this particular AMBN protein, which in other online datasets, not our dataset, the one we use, but in other datasets, it has shown to have up to 25% percentage in Filipino populations, so modern humans from the Philippines. And unfortunately, we did not have Filipino people in our dataset. This protein I highlight because this protein has been used in multiple studies now to identify samples in the fossil record that have it and identify them as being denisovans or belonging to denisovans. And most recently, there was this Homo erectus study that showed actually some of the Homo erectus samples also have this denisovan AMBN version. Finally, we wanted to see what happens in our iterative analysis if we are only allowed to use modern humans that have traditionally very little admixture from other archaics. So these are humans of sub-Saharan African ancestry. And here is a quick plot showing why we use them. So the theory says that modern humans exited Africa a couple of dozens of thousands of years ago. And as people exited out of Africa, so these are the out of Africa populations, they met Neanderthals. So all individuals outside of Africa should have some Neanderthal ancestry. And then some individuals in Asia also met Denisovans, so they have some Denisovan ancestry. However, these original lineage that stayed in Africa did not encounter these other archaic humans, so they should be free of, or close to free of Neanderthal and Denisovan admixture. So initially, we redid our iterative analysis with these 12 novel proteins, and we saw that there was a slight increase in topology in the proteins in the tree supporting topology number 1. So this is when only using African samples for modern humans, and this is when using global humans. And you can see when using 12 proteins, there is a slight bump of this purple topology that has Neanderthals and Denisovans closer and modern humans as being the other lineage. And we also applied the same trick into our dentin dataset, so with the 28 proteins. And again, we saw a slight increase in this topology, the purple one. So if you remember, it was more like 60% of the blue and 40% of the purple. Now we're quite bang on the 50% tree supporting this topology with Neanderthals and modern humans being closer, and 50% this topology with Neanderthals and Denisovans being closer. But still, we get roughly half of the tree supporting a different topology than the one from our whole genome DNA data. So in conclusion for this analysis of assessing the effect of admixture, we saw that there are dentin, bone, and enamel proteins in modern humans that actually likely originate from archaic humans like Neanderthals and Denisovans. And these can have a small effect in the phylogenetic tree you're generating, whether you're using the introgressed humans. However, it doesn't seem to have as big as an effect as other things, like most likely incomplete lineage sorting. Or it could be the case that there are other admixtures of the past that could be affecting our results that we cannot control. Like, for example, we know there was an admixture between modern humans and Neanderthals that happened around 400,000 years ago. And also, there are a lot of papers support that Denisovans have had a admixture with a ghost lineage or a Homo erectus or something other archaic. We don't know exactly when. But either of these two admixtures happening very a long time ago could possibly affect our results and seeing like humans and Neanderthals being closer and then Denisovans being the outgroup. But unfortunately, there is no DNA data to be able to check for this. So in conclusion for the whole chapter or publication, these deep time proteins show different levels of conservation depending on which one you look at, and thus have a different level of phylogenetic information. When you're using protein and DNA data of the same exact locus or same gene, they can support different topologies. The protein trees can differ from the original topology of the DNA tree. And then these 12 proteins can confidently recreate the hominid tree, so between chimpanzees, gorillas, and humans. However, the same 12, or even adding up to 28 proteins, we cannot confidently recreate the late hominid tree between Neanderthals, modern humans, and Denisovans. And this is in small part due to integration between these groups, but mostly due to incompleteness order and how close this population splits between these three groups are in time. And with that, I would like to thank all of my supervisors that contributed for this project. So Fernando Racimo and Enrico Pellini were my original PhD supervisors when we started this project, and Lauritz is my current supervisor and contributed for the latter part of the analysis. And with that, I would like to thank you for your attention and for your time, and I will be happy to take any questions now or at any point if you want to send me an email or anything like this. And yeah, I'm going to stop sharing my screen right now.
Thank you, Ioannis. That was a great talk. So interesting. Yeah, there are already questions in the chat. Can you see it, or do you want me to read the question?
Okay, so the first question, thanks for the entropy calculation. What do you do for positions that are variable within a species? Collagen A1, position 2. So I cannot speak for the specific site, but obviously there are multiple proteins that have some variability within a specific species. So for our iterative analysis, what we try to do is every time if you remember, we did the tree 1,000 times for each number of proteins. What we did was every time we were picking a sample, either a human, or, a chimpanzee, or a gorilla, it wasn't the same human every time. We had the data set for humans, we have more than 100. I think for chimpanzees, around 30, gorillas around 20, and orangutans around 20 again. So every time we redid the analysis, it was a different human. So we expect essentially to incorporate some of that within species diversity into our analysis. Same as when we were calculating the entropy and evolutionary rates, we're swapping individuals every time we rerun the analysis. So we get a rough estimate of how it varies between the different members of a species. For the phylogenetic analysis, it is, as I said, with integration, you should probably pay a bit of attention to try to figure out which human or which member of the species you want to use as a representative for your phylogenetic analysis, because it could affect the analysis. I hope this answers the question. Okay.
If there are any other questions, you can just open your mic and ask it, or you can put it in the chat. Yes, Celine, you can go ahead. Yeah.
Yeah, thank you. Thank you very much for your presentation. I just have one question. I'm sorry I arrived a little bit late, so maybe you said it before, but I was wondering, why do you need to compute a new matrix to kind of understand the diversity, the genetic diversity that is within your proteins? Because I guess if you compute just the average genetic distances for each protein, you should get more or less the same result.
Yes, so for that, my main thought of why I chose also entropy in the beginning is because you have these positions where there are distances. There are multiple positions where you have a different amino acid for different species. You could have 4 different amino acids, and if you calculate the distance, it would be 1 distance between each species. But actually, this position is super variable if you look at the whole clade of species for that position, because there are 4 different positions that can happen, different mutations. So I wanted to incorporate this idea that there are these positions where they are super variable and you can get a lot of phylogenetic information using them. And also, it could be the case that, how you calculate the genetic distance with amino acids is a bit more different, because some amino acids, they just require 1 mutation at the DNA level, but it might be other cases where you need to have 2 different mutations happening on the same triplet to produce a different amino acid. And I think the evolutionary rates, at least that's what it promises a tool to incorporate that type of distance. So it's not the same distance if it's just 1 amino acid position difference. It depends on which amino acid you're comparing to which other amino acid. And for this, I think like the evolutionary uses like a distance matrix, but essentially it is a distance metric as well. Yeah.
Thank you.
Oh, thank you.
Any other question? Yes, Celine?
I can try another one. Yeah. At the end of your talk, you're trying to see the effect of incomplete linear sorting to see if you can reconstruct your phylogenies. But if you can't, could it be just as well that you don't have enough information?
Yes, so in the paper we had this concept that we tried to describe as apparent incomplete linear sorting. So we tried to differentiate when there's like real incomplete linear sorting, like I showed at some point, I don't know if you caught it, had some like figures showing what incomplete linear sorting looks like. But we also term this apparent incomplete linear sorting, which is when you have, because of the drop of information when you're going from DNA to proteins, it would appear as if 2 species are more closely related than what actually the DNA data shows you. And I think when we did the DNA trees versus the protein trees for the same gene, I think this is what we show, is that it could be the underlying gene actually is fine, like it doesn't have ILS, but when you're generating the protein tree, because you lose a lot of information, it looks as if there is ILS. So I use the term ILS here, but it essentially also means this apparent ILS, what we call in the paper, which is, where you don't have information, or like the information you do have gives you a different picture.
Okay,
But
then
thank you for the question, yes.
I understand better. Thank you.
Well, thank you for the comment.
Okay, any more questions? I wait a few minutes if someone is typing in the chat.
Yeah, and I should say that, if you have other questions at any point, I'm happy to talk about this. I dedicated most of my PhD trying to make sense of this type of thing, so I'm more than happy to talk about my experience. Yeah.
This was a very interesting talk, and I think much needed for the future of paleoproteomics and the study of hominins. So thank you again, Ioannis. Yeah, I think if there is no further questions, either people can contact you directly. There's also the paper that everyone can read and go back to. And I will stop the recording.